Professor Tedder, I'm going to pick up the chronology of events in relation to HIV screening in November 1984. By looking at a document with you, it's PRSE 0004191. If we look at the top of the page, we can see this is described as Report on Meeting of Advisory Group on AIDS, 27th of November 1984. This report doesn't have a name attached to it, but it looks like it was probably produced by Dr. Bell, who was an observer from the Scottish Home and Health Department, at the meeting that took place on the 27th of November 1984. Sir, for your note and for the record, I don't have a reference number for the formal minutes of the meeting, but the agenda and details of membership, including Professor Tedder, can be found at CBLA 0011985.
You just give me a moment. There is a, I don't know if you've got reference DHSC 302551_011.
I'm not sure whether we have it on the system today.
Because that's a note, certainly, a very long note of what happened at the meeting, or report of what happened at the meeting.
Could you give the URL again, sir?
Yes, DHSC 302251_011. I can't tell you whether it's minutes or another report. I suspect it's another
apparently
report.
We do have it. Yes, this is Dr. Abraham's note to Dr. Harris.
Thank you.
Of the meeting. In fact, I'm going to go back, if I may, to the document at PRSE 004191, just because there's a comment in there that I wanted to ask Dr. Tedder about, Professor Tedder about. So if we go to the second page, and bearing in mind, of course, this is somebody else's comments on the meeting, Professor Tedder, but we have the paragraph D, development of HTLV3 test facility. Vice now has an isolating cell line which produces virus suitable for assay. Tedder has used this antigen successfully for 2 weeks. This has no licensing problems. DHSS has been informed by US DHSS that access to Gallo isolates and cells can only be via the manufacturers to whom these are already licensed. And then this. Vice Tedder DHSS appeared to be negotiating as follows. Welcome interested, Celtec no interest, Unilever / Seward ? Now, we know from your statement and other documents that Welcome obviously was approached to assist with the scaling up, the commercialisation effectively of the test. Before I ask you anything further about Welcome, do you recall any interactions either that you had or Professor Vice had or that you know anyone else had with any of the other companies named there?
To be quite truthful, no, I don't. I think it's probably unlikely that I would have been involved with that because, as I say, this fell under the remit of Professor Weiss and the Chester Beatty Laboratory. I should have known about it, but I wasn't directly involved with putting it forward as an area of commercialization because it was not my remit.
Now, in terms of the collaboration with Welcome, and we'll see further documents that refer to that over the coming months, do you have any recollection now as to how that came about, whether the department initiated it, or you did, or Professor Vice?
I think it would have probably come about through the relationship which our department in the Middlesex Hospital Medical School had with Welcome in relation to hepatitis B diagnostics. Their particle agglutination test was something which David Dane was very interested in and used, and we adapted that. And as one of the British manufacturers of diagnostics, Welcome would have been the people who Chester Beatty would have gone to. probably on our recommendation because we knew people there, but I was not directly involved with that.
Now, if we then move to December 1984, HCDO 40394_117, please, Lawrence. These are the notes of the meeting that took place at BPL on the 10th of December 1984. We looked yesterday at an advisory document that emerged out of this meeting, but these are the notes taken, I think, by Mr. Pettit from BPL of this meeting, which involved, as you'll see, a wide range of people, reference centre directors, Dr. Krask, Dr. Mortimer, and others, and of course then yourself. If we look at the bottom of the page, please. In relation to HTLV3 antibody screening, it records Dr. Tedder reviewed the current situation by saying that the Gallo cell line was available for investigation, although the US had made isolates difficult to obtain. The British isolate required an organisation to handle the bulk virus culture. Porton and Wellcome are the only ones so far interested. There are problems in obtaining the antigen. Dr. Tedder's test uses a cruder antigen. Do you know what that refers to? The problems in obtaining the antigen, and then a reference to yours.
I can surmise what it was due to. I can't give you gospel because I don't know exactly what they're saying. The difficulty was that you had to have a protocol which allowed you to have expanded cells and then infect them, and then take the supernatant, the fluid from around the cells, and use that diagnostically. And if you didn't do that, and you tried the more conventional way of having infected cells and expanding infected cells, I think we touched on this yesterday, the antigen was not shed by the infected cells into the supernatant the same way as if you had a lot of cells, put the virus in, and then harvest it. And that was the difficulty. And I'm not sure why the comment is a cruder antigen. It may be that we used an antigen which didn't have to be purified. And I think that was the advantage, because if you recall from yesterday, I was saying we had an antibody on the solid phase, which then pulled down the antigen, so you actually did your purification on the solid phase rather than having to do it in a more conventional way.
And then, just to go down the page, under the heading Availability of Tests, it records, Dr. Krask advised that currently the reagents were only available on a research basis and that substantial resources would be required to enable the proposed workload to be undertaken. And then there's a reference to routine testing in the next paragraph. And then this, some discussion took place on which organisation would be best placed to organise the testing and whether DHSS financial support would be forthcoming. Dr. Lane suggested that if resources were available, BPL would play a part coordinating the endeavour. Dr. Smithers, who is the successor to Dr. Diana Wolfert, advised that she would take all these points back to the DHSS for consideration. And then the next page, under the heading Blood Donor Testing, it was suggested that the testing of donors requires either one, mass commercialisation of a British test, or two, application of a current commercial test. Confirmed that testing would be introduced at 2 centres early in 1985 prior to widening availability to the rest of the NBTS. Dr. Gunson advised it would be preferable to test all donors. However, if resources were limited, it might be better to concentrate testing at the major risk centres. And then this, Dr. Cash was concerned that no central organising body was being contemplated for the test programme. This view was confirmed by Dr. Tedder, who was concerned that the pace of test advancement was so fast that the scientists were left to introduce a test as soon as possible. There was also considerable concern expressed over the lack of financial support from the DHSS. Now, I think the latter point, in terms of lack of financial support, it's obvious from this note what's meant by that. Can you assist us in understanding the concern about the absence of a central organising body and the comment that's attributed to you about the pace of test advancement?
It was not something that I was aware of the concern of that. I mean, I think Dr. Cash was wanting to have his organisation to have a significant role in this, and I think that may have been one of the feelings that he was saying. Well, you know, who's going to do it? Why don't we do it? Whoever was going to do it at that time, there would have been a need to use cultured antigen, which means that you have to have the ability to handle what is called a category 3 pathogen under controlled conditions to make sure that it was safe. And that would have required considerable investment in a laboratory to be able to expand that. That I think is indirectly why we talked to colleagues at Porton at one stage.
And then following this meeting, just over a week later, there's a letter from you to Dr. Smithers, PRSE 0001117. This is a letter of the 18th of December 1984, and we can see you make a number of points in the paragraphs of the letter. So one, we urgently need to be able to scale up of the Middlesex Hospital Chester BT radio immunoassay. The MHCB radio immunoassay has been designed to be compatible with the current BTS hepatitis testing. Pilot studies are of the utmost priority in selected centres to confirm this is indeed the case. Until the assay has been routinely used for a considerable time, it's very important that reactive sera are referred to a designated laboratory for confirmatory testing and the donors and their blood products are followed up. And then there's a reference to an initial need to monitor the efficiency with which your assay, if I can put it that way, and the forthcoming commercial kits detect anti-HTLV3. And then there's a request for financial support, so a sum for a basic grade MLSO, a sum for a secretary, and a sum for what's described as predicted disposables. And then over the page, you say, in the third line, we would be unable to take up this work without support, and we would not be able to continue it here at the end of the support period. This work could only have been possible with the cooperation of Professor Weiss and colleagues at Chester BT. Now, if we just go back to the first page, so it's clear you're asking the DHSS to provide what looks like a relatively modest amount of financial support to enable work to continue. But the reference there is to scaling up of the RIA. And as I understand it from your statement, you're slightly perplexed by that because by this time, your understanding would be that scaling up would be something being undertaken essentially by Wellcome. Is that correct? And that they'd be using an ELISA rather than an RIA?
It's difficult for me to recall exactly. At that time, the transfusion services were able to handle radioimmunoassays because the hepatitis B testing was based on radioimmunoassays, so therefore it would not be difficult for them to. They would have the gamma counters, they would have the washing facilities, they'd have the safety of dealing with radio labels. So it would be, from a health and safety executive point of view, it would be relatively easy to give them a radioimmunoassay which would fit to all the hardware that they currently had. I'm not sure that I've answered your question.
So your statement suggests that by this time, if any commercial company was going to be involved, and we've seen the reference to Wellcome, and you've explained that it would need someone like Wellcome to be able to scale up, that you wouldn't be able to do it within your facilities.
Correct.
And Wellcome would be using an ELISA test rather than an RIA.
I think at this time there was a move towards coming away from radioimmunoassays to a more stable platform, which would be an enzyme-linked immunoassay. And if one had, in discussions with colleagues in Wellcome, they would not have entertained running a radioimmunoassay because it was not their expertise. But this is why I think Dr. Lane was anxious to it be recognized that BPL could produce radioimmunoassays for this. But yet the general move was away from an RIA to an EIA, and that would have precluded one working with the BPL, because they did, as I'm looking at it now, I think they were not experienced in making enzyme immunoassay labels and dealing with an EIA version.
If we then just pick up a handful of documents which are departmental documents and not ones you've just seen at the time, but which just show how the department was dealing with the matter. If we start at PRSE 0003287, this is a minute dated the 2nd of January 1985 from Dr. Smithers. It's to the department's STB, so Scientific and Technical Branch, and it refers to a draft paper and a copy of your letter of application, which I anticipate refers to the letter that we've just looked at. And Dr. Smithers says, I hope the proposal will meet with approval. And over the page, we can see what Dr. Smithers here is saying. So an RIA for HTLV3 antibody believed to be a causative agent of AIDS has been developed at the Middlesex Hospital and used there on a research basis. Plans are going ahead to scale up production of the test reagent, and it's hoped that tests for blood donors could be ready to be used in the NBTS in the early part of 1985. In order to monitor the sensitivity and specificity of the test, to validate positive tests, and generally to develop the accuracy of the test, a reference centre for problems experienced by the regional transfusion centres is mandatory. Dr. Tedder, who developed the RIA in collaboration with Professor Weiss, is willing to undertake this function for further development of the test. If he does so, he requires support to cope with the extra work. And then we see the figures that were in your letter. And then the last paragraph, in view of the important role in ensuring the safety of blood donations and limiting the spread of AIDS into the wider community than is currently the case, there is an urgent need to use this test in the NBTS, and its success will depend on its accuracy, which needs to be monitored. Med-SEB, that's Dr. Smithers' branch, see this request as a priority in needing support. So this would appear to suggest that the department's understanding of what you were anticipating your role was going to be was to act as a reference centre for problems experienced by regional transfusion centres in piloting or rolling out the test. Was that your understanding of what you were, the role that you were going to be undertaking?
It's very difficult for me to recall that at this distance. Looking at it now, I would be surprised that I would have been put in this position without the involvement of colleagues in the Public Health Laboratory Service, because they were also expert in the field, and particularly Dr. Mortimer's team already had access to our radioimmunoassay and was using it as one of the tests that he had at his disposal for doing reference work at Colindale. This is very radioimmunoassay oriented, and we already, if this was 1985, we were already thinking that we need to get away from RIA to EIA, and that is why I'm slightly puzzled that we don't have more of reference to enzyme immunoassays with colleagues in Wellcome, because that would have been the natural way to go, because we did not have the experience for making enzyme conjugates, and BPL didn't have the experience for making enzyme conjugates.
If we just then look at a document from a couple of days later, the 4th of January, which is DHSC 0002255_039. If we just go to the third page, first of all, to see who has produced this. It's D.A. Kennedy, who may, I think, have been Dr. Kennedy, but I can check whether I'm correct about that or not. Again, in the scientific and technical branch of the DHSS, 4th of January 85. And if we go back to the first page, we can see what Dr. Kennedy's understanding of the position appears to have been. So if we go to the second paragraph, where it refers to you, Dr. Tedder and Professor Weiss have, with colleagues, developed an RIA for HTLV3 antibodies. This RIA was developed with the intention that it should be suitable for routine use in blood transfusion centres. Accordingly, the basic test protocol is identical to that of an RIA that's widely used for screening for hepatitis B surface antigen, and the same range of equipment is used.
That is incorrect.
In what respects?
The basic test protocol is not identical to that of an RIA used for screening hepatitis B surface antigen. The hepatitis B surface antigen is a two-step immunometric assay which pulls the antigen onto the solid phase and then detects the antigen with a radio label. That is a very different format from the assay which we're talking about conceptually for HIV, which is, yes, you have a solid phase, it's got an antibody to HIV on it, and you pull the antigen onto there. That then becomes the whole reagent for the assay, that you then use this solid phase with the captured antigen to give you an opportunity for blocking an antibody which sticks onto that antigen with the patient's serum, which is a completely different protocol from the RIA that one's talking here for surface antigen.
Is it, however, correct that the same range of equipment is used? So the second part of the sentence.
If it is for a radioimmunoassay, yes, it would be, ma'am.
And then there's reference to the Middlesex having played a big part in the development of the hepatitis test. And then this anti-serum for the RIA is being produced by Wellcome Reagents Limited in collaboration with Porton Down, the PHLS Centre for Applied Microbiology and Research. This is being done on a costs-only, no-profits basis. There's been a patent application in the name of the Middlesex Hospital and Chester Beatty Laboratory, where Professor Weiss is based, and commercial exploitation is likely. The RIA is thus a product of the cooperation of British science and British industry. There is general agreement that it's the most sensitive RIA for HTLV3 presently available. So again, just pausing there, you've already told us about the involvement of Wellcome, the involvement of Porton Down. Was that correct that it was being done on a costs-only, no-profits basis?
I can't comment on that because this would have been agreed with the Chester Beatty Laboratories, and I had no involvement with that. I think there would have been a general feeling from colleagues in PHLS and elsewhere that this was so urgent that this just needed to be done and cover costs and get on with it.
And then the next paragraph begins by saying that there are firm plans to produce the RIA in kit form, the same form as the hepatitis test at BPL, but however work to scale up production will be needed before routine supply to BTCs can be started. So again, just pausing there, is that right, given that the scale up and the production of the test kits, as I understand it, if they're going to be done by Wellcome, would not be in the RIA form, but would be in the EIA form? I
think that is correct, because I do not recall Wellcome having any experience in handling radioisotopes.
There's then a reference to pilot studies on use and performance will have to be undertaken, and throughout these studies, the Middlesex Hospital will have to monitor the results, and then there's some more detail as to what that might set out, what that might entail. If we go over the page, and picking it up in the third line, commercial kits for HTLV3 are expected soon from the USA. It's been predicted these will cost between £1 and £2 per test. This is considerably more than the Middlesex Hospital Chester Beatty RIA will cost. It's likely that USA manufacturers will wish to use the UK as a proving ground for their products and thereby to gain support for performance submissions to the US Food and Drugs Administration. It's very important, therefore, to be able to assess these kits to ensure that the NHS can be told about unsatisfactory ones. Clearly, the Middlesex Hospital is uniquely qualified to assess HTLV3 kits. And then there's a reference to a proposal from you and Professor Weiss, which is I think we may have somewhere, but isn't on this document. And then it says this: after it was submitted, discussions were held with Dr. Tedder to clarify some of the points made. As a result, some changes to the costings can be made and more information can be given. Now, I don't propose to read through all that, but we can see it's a request for 2-year funding, not 1-year funding, and then there's some equipment and consumable items and so on referred to. And then point 6 says, given that funds are made available, it should be possible to start the pilot studies by June 1985. And then if we just go to the next page, the proposal, which is strongly supported by Medical Division and STB3A, offers the opportunity to develop further a very sensitive British test for HTLV3 antibodies and to establish it for routine screening of blood donors for AIDS. The proposal also offers the opportunity to follow up in-depth donations of blood from HTLV3 antibody-positive people. Finally, there is an opportunity to assess commercial products which will inevitably be introduced to capitalise on an established need. Now, do you have any general comments about what's set out here, Professor Tedder?
No, ma'am, I don't. I mean, I think it was, in the end of the day, the move to EIA opened up a very successful collaboration with colleagues in Wellcome Diagnostics, and this was generally recognised to be a major step forward for British industry, as you know. So,
would it be, this document appears to suggest that at this point in time, 2 things were, amongst others, were contemplated. First, that the Middlesex Hospital would be involved in the production of kits that would be used for pilot studies. And secondly, that the Middlesex Hospital would be used or involved in assessing the kits more widely. Was that correct?
I think we would not have been involved with the EIA development because we did not have the technology for that. We would have advised and helped with the generation of antigen or characterization of the antigen, and we would have certainly been involved in dissecting the meaning of a reactive sample in any assay in terms of whether the reactivity was specific for HIV, as we can call it now, or was not a specific reaction.
And then if we just move on in January to a document produced by Dr. Smithers, DHSC 40562. It's 11th of January 1985. It's Dr. Smithers to Dr. Alderslade, and she's sending this so that it can be submitted to the Chief Medical Officer. We can see that from the first paragraph. CMO wished to consider this submission, prepared with administrative colleagues, for ministers to obtain approval in principle for the introduction of a screening test for AIDS antibodies in the NBTS. The UK test is currently being used at the Middlesex Hospital and at Colindale, a scale-up of production of the reagents necessary before the test can be applied more widely. And then if we go over the page, we've got the text of the submission. I don't need to read through any of that, but if we could go to the bottom of the 3rd page. Just under the heading Financial Implications, the submission says this: no tests are yet available for use in regional transfusion centres. They're expected to be ready in the spring. Both American and British tests are still being developed, but the likely cost will be between 75 pence to £2 for each donation. The British test is more sensitive and more suitable to install at transfusion centres and is likely to be cheaper. And then there's a reference to what the resource implications may be. Now, can I just invite your observations on the suggestion there that the British test is more sensitive and more suitable to install at transfusion centres? Would that be correct if Dr. Smithers is talking about your RIA to start with?
I'm not sure what, in this particular point in time, the British test refers to. I do not think it would be the RIA because there was no desire to build an RIA, although we had the BPL RIA for hepatitis B surface antigen. That was at a time when there was a move away from the RIA to an EIA. I'm not sure if they are talking about the competitive enzyme immunoassay as the British test, which I think is where we would have been here, not talking about a competitive radioimmunoassay, but a competitive EIA. I think indeed it was more sensitive, but it was, or as sensitive, but it was very much more specific, which had considerable implications, because if you have a test which has false specificity at a measurable and easily detectable level, every donor who's picked up has to go through a rigorous investigation, not only of the donor, who they are, what they are, their behavior, etc., but also you have to have all the samples have to be put through confirmatory testing to make absolutely certain that what you're detecting is genuinely antibody against, in this case, HTLV3. So the competitive EIA, or indeed if it had been the RAA, but I don't think it was, the competitive assay would have been much more specific. I'm not sure it was necessarily more sensitive than the American indirect immunoassay, probably similar. It had the advantage that it would detect. Can I just go into a little bit of immunology, ma'am? Antibodies can be IgG, IgA, and IgM. The American assay used an anti-IgG detector, so would only detect IgG antibody. A competitive assay will react with and detect antibody of any specificity, because any class specificity, because that antibody will get in and block the available epitopes, the available antigen sites for the conjugate to come in. So you have an advantage of sensitivity to all types of IgG, IgA, and IgM, which is an advantage. It was easier to use because it was a one-step assay. Basically, you have your well, you put your sample in, you put your conjugate on, you set it incubating, you go away, you come back after the incubation period, wash it and develop it. Whereas the indirect immunoassays, you have to do your dilution, you put it in, you incubate it, you wash it, then you put the conjugate in, incubate it, wash it, and then come to the color. So there are two steps in a sequential indirect immunoassay, whereas the one-step assay is everything goes into the well, incubate, and then wash and develop.
As I understand it, at this point in time, January 1985, the American kits had not yet been evaluated. It's suggested here they're still being developed. So in terms of an ability to say that the British test, and assuming that's a reference to the British EIA that was being developed with Wellcome, would have greater specificity, would that be based upon an understanding of the advantages of the competitive assay over the indirect assay in general?
Well, that would be an aspiration. I suspect my colleagues in the PHLS would have done work on this to also look at the sensitivity of the competitive assay. It would have been easier to install in the sense that you just have one incubation and then one wash and develop. You don't have to have two sequential incubations, which makes it technically easier.
And just for the sake of completeness, there's an annex to this submission which begins on page 5. And paragraph 3, bottom of the page, has a heading, Development of the Screening Test for AIDS Antibodies. And there's reference there to the 5 pharmaceutical companies in the US who've been licensed, and then reference to Professor Weiss having isolated a virus from a British patient, and that this was being developed to provide a test by Wellcome, who subcontracted Horton to scale up its production. And then if we go over the page, we look at the last paragraph on this page. It says this: the UK test is sensitive and specific, and particularly appropriate to introduce into RTCs who are using a similar technology to detect hepatitis B carriers. Now, that would appear to suggest there's still an understanding on the part of the department that it's going to be an RIA test rather than an EIA test.
I think you're right. What I don't know is when there was a move away from the BPL RIA for hepatitis B surface antigen to a commercially available assay for enzyme immunoassay for hepatitis B surface antigen.
Excuse me. If we then just move to the end of January 1985 and the first meeting of the Expert Advisory Group on AIDS, of which you were a member, that's at PRSE 30...
I'm sorry for interrupting, but can I just be clear? If we go back to the previous page that we were looking at, page 3 of this memo, thank you. What it says...
So that's page 5.
Right, page 3, it's the third page of the original, page 3003, thank you. Under 6, the fourth line, British test is more sensitive and more suitable to install at transfusion centres. On the face of it, if one's asking, is what is in the mind of the department at this stage RIA or EIA, this is ambiguous because the sensitivity of the test does not depend upon whether it's RIA or EIA. That's plain from your evidence. And you're saying, well, it is more suitable to install because it involves a simple one-step process as opposed to the American two-step process. So you could say it's more suitable to install. So that language doesn't help with knowing precisely what's in the mind of the author. But if we go then, please, to where we were, page 5. Yeah, next, thank you.
I think it's probably the next page.
It's the next page, thank you. Where it talks about, yes, it's the last paragraph on the page. The UK test is sensitive and specific, and particularly appropriate to introduce into RCCs who are using a similar technology to detect hepatitis B carriers. Now, you have said to Council you think that must be, in context, a reference to the RIA process and not to the EIA.
Correct.
And so probably it would follow, if that's what's in the mind of the author here, that the earlier ambiguity is resolved. It's looking at RIA and not at EIA. Would that be fair?
I think the comment on sensitivity and specificity and the ease of a one-step assay is applicable both to having a radioimmunoassay label and having an enzyme label. The overriding issue is what platform the transfusion service had at the time, and if they're using a radioimmunoassay, an RIA competitive assay would fit very well. If they were moving to an EIA, a one-step competitive EIA would also fit. So it depends on tailoring the need or tailoring the feature of the assay to fit with the need of the transfusion service and whatever their infrastructure was at that time. So in either way, the sensitivity and the specificity as an RI was similar to the sensitivity and specificity of an EI in competitive format.
Yes, one of the questions for me will be what was in the mind of government at this time, and was it a justified view? That, of course, we've had some evidence, oral evidence, but will largely be a question of interpretation of, or may well largely be a question of interpretation of documents. I think by this stage, which is the mid-January, 11th of January, there isn't a document coming from the department which refers in terms to an EIA test being that which is linked in the writer's or author's mind with the British test.
I think that's right. And there's the document we're about to look at, which are the EAGA minutes, does start talking about ELISA tests for the first time, but it seems to be referring to that as being what the American companies are producing. But again, it's all a matter of inference from documentation.
So, but it does look that that's the mindset at any rate. Rightly or wrongly.
Yes.
And for what you're saying, they may not have picked up with the latest developments in the trends of the tests which virologists would provide.
I can't comment on that.
No,
I'm not in their mindset. So, but yes, I mean, I think that's possible. It depends where you perceive the technology of testing is moving, and at this time it was clearly the American assays were using chemiluminescence, which is an emission system, which is more like a radioimmunoassay, except in this case it's emitting photons. And we were using a colorimetric assay as an inhibition assay. So it's slightly different, but they're all forms of enzyme-linked assays.
Thank you.
And so if we move to the end of January and the first EAGA meeting, PRSE 0002734, we've got the date of the 29th of January 1985, and we can see it's chaired by Dr. Abrams, and then we have a list of attendees, including Dr. Arteda. And then if we go to page 4, we have the heading, the availability of the AIDS screening test. Paragraph 18 refers to Professor Weiss, saying that work was currently being carried out with Wellcome Diagnostics to develop a screening test, but there were still problems to be solved. He was not able to say when the test would become available. Professor Zuckerman said that tests were also being carried out at his laboratory and that the results of the American Dupont and Travenol tests might be available within a few months. Comparisons will be made with the tests being developed by Professor Weiss and Dr. Tedder. Paragraph 19, the chairman reminded members that the November meeting of the advisory group had concluded that a screening test for all blood donors should be made available as soon as possible. He asked whether the EAGA endorsed this view. There was general support for the introduction of a blood donor screening test as soon as practicable. And then in terms of type of test, we have paragraph 21. On the type of test to be used, Dr. Gunson said there was an overwhelming preference for the use of the radioimmunoassay test in the NBTS, whilst Professor Zuckerman stressed the need first for evaluation of other tests, including the ELISA test. The chairman said that the DHSS would ensure that all tests were evaluated. And then paragraph 22 is about availability of testing for GUM clinics. And then 23 refers to a subgroup being set up, of which you remember, Professor Tedder, to consider various aspects of the screening tests. So it might be thought paragraph 21 indicates an understanding at this meeting that RIA tests were still an option. By this time, end of January 1985, to your mind, would there have been any prospect of an RIA test being used in the National Blood Transfusion Service?
In the sense that the infrastructure was already in place for radioimmunoassays because of the hepatitis B antigen detection requirement, yes, but if you recognize that all the evolving commercial assays for antibody to HIV or HW3, if we may call it HIV because that's what we're talking about, all the anti-HIV assays that were commercially available were EIAs. So even if the NHSBT and Harold Gunson wanted to maintain RIA, he would have not been able to do so on the commercial assays because they were all EIAs. So I think this is, I'm not sure. There may be an overwhelming preference to use RIA because they had that in place, but it was unsustainable in the eventuality because the assays for anti-HIV were all enzyme linked.
And then we can look at the first meeting of the subgroup that's referred to in paragraph 23, It's at DHSC 0000425. So it's a note of a meeting on the 15th of February, and we can see those in attendance, including you, Professor Tedder. And then paragraph 2, it was recorded that Travanol, DuPont, Ortho, Abbott and Electronucleonics were licensed to use the Gallo isolate. Apparently all the US companies were using an ELISA test, none had yet been given FDA approval. Wellcome were developing a test based on the British isolate, which might be available for use in regional transfusion centres within 3 months. Doesn't actually tell us in terms whether that's going to be an ELISA or an RIA. And then under the heading evaluation, the chairman said that in the absence of statutory marketing controls, the DHSS had invited companies developing test kits to take part in a departmental evaluation. So just pausing there, and this is more an observation than a question. It would appear from this that by the 15th of February, the invitation to companies to participate in the evaluation had already been issued. And then it says this, an ad hoc panel of experts with DHSS officers would agree a protocol and arrange for a PHLS virologist to carry out the evaluation. And we know that was in due course Dr. Mortimer. The British test under development would be included in any evaluation. Professor Tedder, I just wanted to ask you this. In terms of agreeing a protocol for the evaluation, how long broadly would you expect that to take? Would that be a difficult task, or would that be a relatively straightforward task?
Can you differentiate between the setting up the structure for an evaluation from the setting up the access to the antibody assay? Because I think those are slightly different. The development of a competitive ELISA by Wellcome Diagnostics would require them to make the components, get access to a supply of HIV antigen from culture, which in the very earliest stages would have been through collaboration with the Chester Beatty laboratories, but very quickly would have had to fall to the Portman group to produce it because of the containment you needed to grow up large quantities of a category 3 pathogen. I'm not sure how quickly I was expecting anything to happen. I was probably pretty busy providing serological testing for clinics and for, as you know, for haemophilia centre directors at this time, and I was we were pretty much pushed to limits in providing that serology, so I would not have been close to Wellcome at that point in time, but I'd been asking them to expedite everything as fast as they can.
And would it be right to understand that at this point in time, so mid-February 1985, Wellcome were not yet at the stage that the American companies were at?
I think that has to be correct, because the American companies were saying, well, we have assays which you can purchase and they're ready for use. They were indirect immunoassays of questionable specificity.
And then just going back to the reference to a protocol, that I think is the protocol for the evaluation rather than describing what it was that Wellcome and the others.
I think that's correct.
Would you expect it to take long for a panel of experts with DHS officers to agree a protocol, or should that have been a relatively straightforward exercise?
Well, you know what panels of experts are like. There's a lot of discussion. I think it would be relatively easy, particularly in collaboration with colleagues in Colindale, to define what would be a suitable panel. The difficulty was to have a sufficiently large volume of the start material to be able to divide it into into panels of samples for people who are interested in having access to a panel. And that's the role of NIBSAC, such an important role that they have.
Going to move then to March 1985. And could we have on screen, please, USOT 5016_144. Now, if we look at the top of the page, we can see this is a meeting between CMO, so that's Donald Aitchison, and Dr. Tedder, Professor Weiss and Professor Adler, Middlesex Hospital, 22nd of March 1985. And this document's divided into two stages. There's a note of a meeting with you and Professor Weiss, and then there's a note of a meeting with Professor Adler, and I don't need to ask anything about the meeting with Professor Adler. It says this: the availability of antibody screening tests was discussed. A number of companies from the United States were entering the market. The screening parameters of these tests were not yet established. Professor Weiss had some reservations about the practicality of mass usage of the confirmatory test, now approved by the FDA in the US. The evaluation of the screening test to be available for use in the United Kingdom was discussed. CMO asked whether a recognisable and organised system for this evaluation had been set up. In reply, it was explained that the PHLS would be responsible for the mechanism of evaluation under the direction of a working group chaired with this responsibility. And then paragraph 3, sera from patients with various defined categories of AIDS presentation would be used to identify tests which appear to perform well. It was to be anticipated that the bulk of the commercially available tests would pass this initial assessment. It would then be necessary for initially successful kits to be evaluated in service in the BTS, preferably for more than the 1,000 tests probably now being proposed. Dr. Philip Mortimer was mentioned as a possible organiser of the evaluation. CMO asked whether there were any practical or financial blocks to this programme being implemented. In reply, it was stated that more staff would certainly be required within the virus reference laboratory with associated revenue consequences. The Tedder Weiss test was discussed. Naturally, they hoped that this would prove to be scientifically and practically acceptable in routine use. At the moment, it worked reasonably well as a laboratory tool, but adequate scaling up was still to be achieved. Some delay with the delivery of the bulk antigen from Porton was being experienced. It was agreed that it would probably be necessary for the BTS to go ahead and use the first successful test that became available. This was unlikely to be the Tedder Weiss test in the first instance. They themselves were essentially laboratory scientists. They must inevitably now leave the bulk of the commercial exploitation to Wellcome and Porton. Excuse me. First of all, do you have any recollection of meeting the Chief Medical Officer on this occasion?
No, I'm afraid at this distance in time, ma'am, I don't.
Now, this would appear to suggest that, again, the US kits are at a more advanced stage than what I'm going to refer to as the British test. It's referred to here as the Tedder Weiss test, but what appears to be being discussed here is the test that you and Professor Weiss had devised, which was now being scaled up by Wellcome and Porton.
A competitive one-step EIA, yes.
And it would appear to be recognised that there was still quite a bit of further work to be done in terms of that exercise, and there's reference to Porton, delays with the delivery of the bulk antigen from Porton. Is that a fair reading?
I think we were disappointed with the first products from our colleagues in Porton because they had not adhered to our protocol for cell expansion, then infection. They had gone a different pathway, and it did not produce an antigen which worked. Or there was insufficient, putting it into correct terms, there was insufficient shedding of virus envelope protein from the cell culture when the cell culture was set up the way that Porton did it, as opposed to the way that we set up the cell culture.
So, if it were to be the case, and this is a hypothetical, if it were to be the case that the evaluation wasn't going to take place until the British test was available, that would mean a delay in the evaluation because the British test, as it would appear from this document, was some way behind, at this stage, the US tests.
I can't comment on that because I was not involved, but I think what you say is correct. It would have been uncomfortable to be associated with a development which was going to lead to a delay in investigation of the performance of other kits. What they're actually referring to in the earlier part of that comment was the Western blot kit as the confirmatory test, which people were really fairly nervous about, and we can explore that if you wish. But I think there should have been, I like to believe there would have been an investigation of the performance of the indirect immunoassay, which is the various American or the Pasteur assays, and determining the sensitivity and specificity of those.
And just for the benefit of those listening, in terms of the evaluation process, although I think that some of the earlier documents anticipate that you might have an involvement in that, there is a document that makes clear that you were not to have an involvement with that, nor was Professor Weiss, because there could be a conflict of interest.
Absolutely.
Or there would be a conflict of interest
There would be.
because you were involved with the development of the Wellcome test.
Correct.
So you yourself were not directly involved in the evaluation process. You were on the subgroup of EAGER, but you weren't undertaking
We
the
would have
evaluation.
probably looked at other people's data and poked it around, but I specifically stood back from that. At the Middlesex Hospital Medical School, we were not involved.
And then, again, just for the benefit of those following the evidence, there is a follow-up minute from Donald Aitchison, which I'll just put on screen briefly. It's USOT 5016_143. Where he says this in the minute to Dr. Abrahams of the 25th of March. I visited Dr. Tedder, Professor Weiss and Professor Adler at the Middlesex Hospital on the 22nd of March 1985. During our discussion, it became clear that unresolved technical challenges facing the UK test mean that it is unlikely to be first in the field. We are likely to need to evaluate a number of other tests, largely from the United States, over the succeeding months. I would be grateful to know what organisation exists within the department and the PHLS to meet this challenge, and who is to be both responsible and accountable for the completion of what will be a demanding series of evaluative tests. I would also like to know what arrangements are planned to put into effect recommendations resulting from the evaluation when they emerge. And then paragraph 4, he says he'd like to take forward urgently work on the resource implications of the introduction of the test and its consequences. So that's the position as at the 22nd and 25th of March 1985. If we then move forward to the end of May 1985, to PRSE 0002837. This is a meeting of the Expert Advisory Group on AIDS on the 29th of May 1985. And we can see again, Dr. Tedder, you're listed there, Professor Tedder. And then if we go over the page, we have the heading at 5, Introduction of a Screening Test for Antibody to the AIDS-Related Virus. I don't think we need to look at paragraph 5.1. Paragraph 5.2 then explains what the position is in terms of evaluation. Dr. Smithers said that the PHLS had been asked to evaluate all available screening test kits. 3 produced in the USA had been licensed by the FDA. There were at least 2 being manufactured in Europe. Dr. Mortimer said that the initial evaluation would be undertaken at Colindale, involving 350 sera, half of which were from blood donors. 2 kits would be tested in the next 2 weeks, a third in the next 4 to 6 weeks. It was hoped that at least 3 sets of data would be available for discussion by an ad hoc group of experts in mid-July. And then the decision continues, the paragraph continues, and then the last sentence of that paragraph, the chairman said that while it was important to introduce a reliable screening test as soon as possible, an effective evaluation of the tests was essential and should not be rushed. Now, this indicates that as at the end of May, the evaluation process by the PHLS had not begun. and it's anticipating it will be undertaken essentially in June and July of 1985. Do you have any knowledge, Professor Tedder, as to why, as at the end of May, the PHLS evaluation had not yet started?
No, I don't. I have no recollection of that. All I would say, looking at it now, the difficulty, not the difficulty, but the requirement for an evaluation like that has two components. It has the evaluation has to look at what the sensitivity is. If you have 100 genuinely, you assume genuinely HIV samples, does the test detect 100? Does it detect 95? And what do other tests do in terms of detecting the real reactivity on the one hand? And that's, if you like the term sensitivity, how many out of the positives does it detect them all, mostly all, some, etc. And the other side of that component is if you have 100 or 200 or something negative samples, does it correctly ascribe those as being negative, or does it describe 1, 2, 3, 5% as being reactive? Because that in itself is, both of those are problems. If you have an insensitive test, you're missing genuine positives. If you have a non-specific test, you're generating positives which are not real, and you have to be able to sort those out, because the individuals, whether it's a donor or somebody in an STD clinic, you're telling them you have a reaction in an EIA, and the next question is, what does that mean? And unless you do a whole range of subsidiary testing, you have no idea what it means.
But are you aware of any reason why the PHLS evaluation could not have begun earlier than June 1985?
Well, I think probably, no, I don't, but I think it would have taken a certain amount of time to gather enough samples that were genuinely positive and enough samples that were genuinely negative, and have those categorized so you have a panel of known probity to put the commercial test or anybody else's test through. Because we're a little bit retentive in terms of data as virologists. We like to make sure we've got it right. And you would have to collect a panel of known positives and a panel of known negatives, and that might take a bit of time.
The paragraph below, so paragraph 5.3, records what I think is a discussion about confirmatory testing. We see the reference there to the Western blot and/or RIPA. What's RIPA referred to?
Well, that would be a radioimmunoprecipitation assay, and I have no experience of those.
And in terms of confirmatory testing, you mentioned the Western blot.
Might it be RIBA?
So,
should it be RIPA or RIBA?
I think it's a PA. RIBA? I'm not. Well, it could be either. It could be both. Recombinant immunoblot assay.
That's what I was thinking of.
One, or a radioimmunoprecipitation assay would be a RIPA. And I'm not sure at this point in time. I'm not sure quite what they meant.
Because RIBA was used quite a bit, was it not?
Yes.
Is it right to understand that the confirmatory procedure that you were I think most familiar with, most confident in, was the indirect immunofluorescence antibody test?
It was one which would be, because of using the immunofluorescent assay for respiratory viruses, it's something which a laboratory, diagnostic virology laboratory would find it easy to deal with, because all you do would be, you would replace the film of influenza-infected cells with a film of HIV-infected cells, and then the procedure would be exactly the same. You put the diluted serum on there, you incubate it, you wash it, and you come in with a conjugate of anti-fluorescent labeled anti-antibody incubate it, wash it, and then put it under a UV microscope.
And was that the test that you'd used for the study that had been reported in the Lancet in September 1984? There were the two tests, your competitive assay and the immunofluorescence test.
Yes, it would have been an indirect immunofluorescence assay.
And so you mentioned, I think, having reservations about the Western blot test. Why was that?
In principle, a Western blot, what it does is it takes recombinant proteins and one way or another it separates them on size or charge and layers them onto a solid phase, so that you have an array of proteins on a solid phase. You then bring the patient's serum and you put that over the solid phase, and the patient's serum, antibody from the patient's serum will bind with various components of the virus, which are laid out by size in the Western blot. The difficulty with Western blot is that many of us who used them felt that there were only two types of Western blot that were useful. One where you had multiplicity of lines, and one where you had no lines at all, so completely negative or completely positive. And the difficulty with Western blot, as some people in the audience may recall, as the virologists here, was you were getting a Western blot with one line positive or two lines which were weakly positive. And how do you relate that to the portfolio of antibody that you see in a real, when I say real, that's an inappropriate term, a known positive sample where you have a multiple stack of lines, you have maybe 4 or 5 lines in the Western blot. When you've only got one line in the Western blot, you have to say, well, I'm not sure quite what that means, especially if it's a weakly reactive. 2 or 3 weak reactions in the Western blot were not uncommon. And one then was a major difficulty of interpretation of that. So it was a Western blot in our book. If it was all positive, yes, there would be no difficulty with that. If it was all negative, that was comforting. But then these frequent findings, or 1 or 2 lines, which seemed to be, depending on which sample you had, you'd have a line there, or a line there, or 2 lines there, as opposed to a whole stack of positives. They were difficult. They were not easy. In fact, they were difficult to interpret, especially when you got these partial reactions.
And do you recall which confirmatory test was in due course used once testing was introduced in October?
Well, I think laboratories in testing would have had a range of assays. They would have had an indirect immunofluorescence assay, as we used. They could have had a competitive assay if they'd used the Welkerzyme, and that was very unlikely to give you a false positive reaction. And I don't know, each laboratory would have had its own protocol for building a range of 2 or 3 assays, and knowing if they're all positive, that's one thing, if they're all negative, that's another, and then puzzling about the idiosyncratic single reactions.
And then there's just 2 further documents to bring the chronology to the point of at the end of the summer 1985. So if we start with DHSC 40551, and if we go to the second page, so we can see this is a meeting on the 10th of June 1985 of the screening test subgroup of the Expert Advisory Group on AIDS, attended by you. And if we go a little further down this page, there is a heading, Progress on Evaluation of Diagnostic Kits for HTLV3 Antibody. I don't think I need to read through paragraphs 4 to 7, but if we go to the next page, and just pick it up at paragraph 9, Dr. Mortimer reported that 3 manufacturers' kits would be tested by the end of June, including the Welcome kit. The protocol could be amended to allow the field trials to go ahead earlier than presently planned. However, Dr. Mortimer had reservations about such action before PHLS had evaluated more tests, including that of Welcome, but appreciated the NBTS position. And then paragraph 11 records 2 regional transfusion centres being particularly anxious to start routine testing in advance of a national commencement date. Paragraph 12 refers to the Western blot test. and there's a record of some suggestions by you and a proposal to consult you and Dr. Mortimer about that.
Sorry, which section
Sorry,
was that?
paragraph 12.
12.
And then it's really paragraph 14 I wanted to ask you about, but I might need to read paragraph 13 to make sense of it. So Dr. Gunson then reported on the protocol for the field trials, and there's a further discussion there set out. And then you're recorded in paragraph 14 as saying this: Dr. Tedder considered the sample size might not produce a single genuine positive. The evaluation was therefore about how to employ in the National Blood Transfusion Service rather than a field trial. Are you able to assist in understanding what that means?
If you're going to do an evaluation, you need to have known reactive, genuinely, I don't like the term, but a genuine positive. You have to have samples which you know have genuinely got antibody in there. You have to have a panel of those, and you have to have a panel of negatives. So I would have been nervous about any transfusion centre introducing these tests until the STD clinics nationally had access to the appropriate serology for their patients, because of the danger of saying we're going to be testing in a transfusion centre for HIV and then drawing people in to get their test by default. I think the processing 580 specimens a day on average, if the prevalence was 1 in 1,000 to 1 in 2,000, you would have to do a large number of those to find the one genuinely seroreactive in your field trial. So that's why looking at what I'm told I said at the time, I think you need to have a big enough sample in a very low prevalence situation to find 1 or 2 genuine positives, rather than, if you can follow my concerns, you have to have a big enough panel to give you a small number of real positives to look at the performance of those in relation to the performance of the negatives.
And in any event, and again, this is an observation rather than a question, it would appear from this that the welcome test is now, by the middle of June, available for evaluation. Last document then is PRSE 302628. This is the Expert Advisory Group on AIDS meeting of the 30th of July 1985. And if we go to page 3, this is really just to complete the sequence of events, Professor Tedder. We've got evaluation of AIDS screening tests, and there's reference to a paper being tabled. Dr. Smith is reporting it will be issued to health authorities as a report on the evaluation of the kits. The kits have been tested against a panel of sera from unselected blood donors from groups of patients with AIDS or AIDS-related diseases and from groups of patients in which false positive results were a possibility. The kits recommended as most suitable for use in diagnostic laboratories were, and then we've got the Organon kit, the Welcome kit and the Ortho kit, and various matters set out in relation to them. And then the Welcome and the Organon kits are considered to be particularly suitable for use in blood transfusion centres, were easy to use. Both these kits would be the first to be investigated in the second stage of the evaluation, which was designed to investigate performance in large-scale screening of blood donors. In terms of that second stage of the evaluation, did you have any involvement in that process? So we've got the evaluation that had been undertaken by Dr. Mortimer and concluded by this point in time, end of July, and then an anticipated second stage, which, as I understand it from this, was expected to look at how things, how the testing actually worked in the regional transfusion centres on a large scale.
It's difficult for me to recall at this point in time. I would be surprised if we were not involved in terms of identifying appropriate positive and negative sera for some of our colleagues to use But at this point in time, I really can't say how much, if we were involved at all in that.
And then just to note, if we look at the bottom of the page, there's a discussion in paragraph 7.3.2, which is the last paragraph, about the timing of introduction of the tests. And the reference there in part is to the issue that you've raised, Professor Tedder, about the risk of people turning up at blood transfusion centres effectively to get an AIDS test. I want to pick that up with you as part of a number of more general questions, but we can note that that's being set out there. So I see the time. Those are all the documents on this issue that I wanted to take Professor Tedder to, but I wonder if I could pick up after the break the handful of additional further questions on this issue that I have.
Yes, well, let's do that and come back at 10 to 12.
Thank you, sir.
10 to 12.